Half a day. My name is Justin Berg. I am currently an EBSCOR graduate research assistant at the University of Guam Marine Lab. Today, I will present about my current thesis topic, Here's the Dirt, Sedimentation Effects on Coral Microbiomes. To get started, I will describe the background of my project. Sedimentation is the process in which soil or rock is transported from its original location. Large tropical disturbances can lead to high erosion and sediment loading. This sedimentation and nearshore runoff are one of the primary threats to marine ecosystems for islands across the Pacific. It is important to note that erosion is a natural process, but anthropogenic effects are rapidly increasing runoff. Of the 100 named rivers and streams on Guam, 46 drain directly into the Pacific Ocean. This includes not only rainwater, but erodible solids such as soils, pesticides, and debris. When these maps overlay, it is easy to see the influence that anthropogenic features have on localized watersheds and drainage systems. It is worth noting the increased anthropogenic effects near shore that may have a larger influence than higher up in the upper watershed area. However, construction is not the only anthropogenic impact on Guam. According to Min in 2006, fires were non-existent prior to human arrival on Guam. Almost all fires are set intentionally to clear vegetation, burn trash, and the illegal hunting of pigs and deer. Chronic fires can lead to exposure of the highly lateritic clay soil that eventually forms badlands, as you can see in the picture here. Plants cannot establish in this area, and the outer edges promote further erosion. This can lead to the replacement of forests by non-native savanna grasslands that are more resilient to fires. Fires exist largely in southern Guam. The south gets hit harder for multiple reasons, including types of forest, grasslands, soil types, and topography. In 2019, Guam had less fires than an average year, but they burned 191% more acres than an average year, with 7.1% of the island burned. I want to emphasize that roughly 7% of the island was unnaturally burned. So, what does that do to corals downstream? Terrestrial runoff or wave resuspension of deposits can expose corals to nutrient-rich sediment. Increased eutrophication and sedimentation affect coastal coral reefs worldwide. Areas with coastal development are common zones with sediment that cover reef organisms after flood plumes and resuspension events. An example is Fuha Bay in the late 1980s with the construction runoff from Route 1. Nutrients in rivers from fertilizers, sewage, and eroding soils have increased globally since pre-industrial times, affecting about 25% of coral reefs around the world. While sedimentation can affect coral cover and diversity, what happens on the microbial level? A coral consists of a complex holobiont that is comprised of the host coral and a microbial community. In this study, I am studying the bacteria, specifically the microorganisms and their genetic material that represent the host microbiome and their metabolic adaptations to local environmental conditions. While there may be shifts in corals, it is important to note what aspect of the microbiome it affects. The core microbiome consists of the bacteria that are shared among coral communities. They aid with how host microbiome interactions are established and maintained. They are needed regardless of the environment and play key metabolic functions. The dynamic or variable microbiome stem from the environment, the coral species, and seasonality. They have the ability to acclimate when facing environmental changes. Ziegler et al. Discussed the idea of microbial conformers and regulators. I aim to better understand our study coral's microbial role across site and season. These shifts could have implications for restoration and resilience. What exactly do I mean by this? Stability is referring to the gain or loss of diversity in bacterial species in the coral microbiome. A more stable coral microbiome tends to have higher resilience, according to Bertoli et al. 2018. A more stable microbiome follows the insurance hypothesis. Microbial diversity stabilizes microbial community function. Function could be deemed more important than taxonomic diversity, as multiple species could have the same functions. While taxonomic composition may shift across the environmental gradient, their function may be conserved within the new taxonomic groups. With this understanding, we can now dive into my site location and coral species. For this study, Fuha Bay was chosen as the study site. The Lasafua River drains into Fuha Bay in southwestern Guam. This site has been highly studied in the past in relation to coral composition and sedimentation. This site has also been studied for coral diversity as far back as the 1970s. This site experiences Guam's wet and dry season with an average rainfall of 2.5 meters. My study species is Parietes lobata. This reef-building coral is one of the few species that can survive the harsh conditions from sedimentation that tracks along the inner to outer reef gradient. In this photo, you can see my two sites located at the inner and outer bay. For this study, 128 coral samples were collected. 8 samples were collected monthly from the inner and outer site. The 8-month study was broken down into a 3-month wet season and into the 5-month dry season period. This allowed for a temporal gradient during the study. Once collected, the samples were frozen in liquid nitrogen on the boat. The parent colonies that graciously donated our samples did recover quickly with minimum damage by the end of the study. Coral tissue was isolated from the skeleton via a mini bead beater. Total DNA was extracted via a DNA Easy Power Soil kit in conjunction with the QIAcube Connect. This was quantified with a Quibbit. The V4 hypervariable region of the 16S rRNA was amplified with the 515F 806R universal primers. We ran the DNA through the 16S rRNA Illumina NovaSeq at CD Genomics in Long Island, New York. Of the 128 original samples collected, 114 were successfully sequenced with CD Genomics. After reads were filtered on QIIME 2.0, 112 samples were kept and rarefied at the 60,000 read mark. On this slide, we have 6 forms of environmental data that correspond with Fuha Bay and localized river data. On the top left, you can see the temperatures of my inner and outer site and the drop from the wet season down into the dry season. While the wet season historically has ended in December-January, in 2019, the transition was closer to November-December. The average monthly pHs stayed consistently apart with the inner site staying higher. Lastly, the monthly conductivity shifted with the outer site being higher until the shift to the dry season when the inner site took over. The 3 bottom graphs all represent average daily rain totals, monthly discharge in the Lhasa Fua River, and the monthly gauge reports in the Lhasa Fua River. These 3 graphs all reinforce the shift of the wet to the dry season in Fuha Bay. While this environmental data shows shifts between site and seasons, what is happening microbially? On this slide, we have the taxa bar plot broken down into site and season groups at the phylum level. It can be seen that Proteobacteria, the large yellow group, dominates the Parietes lobata microbiome across most samples. At the phylum level, it is worth noting the increased diversity on the inner wet season. On this slide, we have the taxa bar plot broken down into site and season groups at the genera level. In this graph, the orange group of the family, Endozoicoinaceae, dominates most of the coral samples. This family is known to be nitrogen fixers. The lavender group is from the genera Herbospirillum. This species is a soil-related bacteria. Again, it is worth noting the increased diversity in the inner wet corals. On this slide, I will show the Shannon diversity of the inner and outer sites across the 8-month period in the wet and dry season. This Shannon diversity figure shows the richness and evenness of the samples. In this specific figure, you can see that the inner wet group has the highest Shannon diversity indices and drops when entering the dry season. It is worth noting that the first month in the outer wet season group may be higher than usual due to a typhoon that hit just prior to the start of this experiment. For this unweighted unifrac PCOA, I chose to use this distance matrix based on the presence-absence without abundance as one taxonomic group would have skewed all data, and the unweighted unifrac's incorporation of phylogenetic distances as well. In this figure, the blue indicates the wet season and red indicates the dry season. The filled-in circles represent the inner sites while the outer circles represent the outer sites. Lastly, the size of the circle represents the individual Shannon diversity from the slide before. The inner wet group clusters on the right in a unique grouping that has high Shannon diversity indices. After running a permanova on the site season, the groups are significantly different with a p-value of 0.001 and a pseudo-F value of 5.42. On this slide, I am looking at the anisim analysis. On the left, it shows that the inner wet distance to the centroid is lower than the inner wet's distance to the other 3 site seasons. The p-value of the anisim is 0.001 with an R value of 0.42. The R value shows that there is some difference with some overlap as well. This is expected as I do have some overlaps with site and season as well as 4 unique site-season combinations. After completing the Adonis test, 5.7% of the overall microbial diversity could be attested to site, while season attested to 3.4% of the overall microbial diversity. When the various combinations of site and seasons are considered, 13.1% of the overall microbial diversity can be attributed to this combined variable. Next, I will show the microbial heat map across the site and season. As mentioned across the other diversity indices, the inner wet group is the most diverse. Not only does this group have the highest diversity, but they also have some of the highest abundances. One specific taxa to look at is the Endozoicomonaceae, highlighted with the yellow line near the top. This is a dominant group across all taxa and is associated with nitrogen and carbon fixation as discussed before. While the heat map listed the 30 most abundant taxa across site and season, I also broke down the core microbial taxa by site, site and season, and the inner wet group. It is worth noting that the wet season has more core taxa despite location, while the sites tend to be more similar despite time. When comparing site and season, the inner wet holds the most core taxa compared to the other three site-season combinations. When comparing the three inner wet months, the October 22nd time point has the most individual core microbes. It is also worth noting that the core microbe numbers increased in the wet season through time. For this study, we came up with three conclusions. The first, site or season are not the largest influence, rather site and season, specifically the inner wet. The second conclusion was that the microbial diversity of site and season shift through time and space. And lastly, while bacteria do not dictate coral diversity and coral cover by themselves, the instability of the bacterial microbiome likely contributes to decreased coral cover and diversity in Inner Fuha Bay. Lastly, what's next? We'd like to examine the core taxa in pie crust to test the coral insurance hypothesis to see if microbial functions are conserved through time and space. First and foremost, I would like to thank my co-author, Dr. Bastian Bentlage. I'd like to thank James Pfeiffer and the Davies Lab, my committee members Dr. Peter Hauck and Dr. Larry Raimundo, Claire Moreland Ochoa for her constant support, Guam EPSCoR, NSF, and the University of Guam. With that, I'm open to any questions. Thank you.